Welcome to the continuation of Lecture 5 video supplements. This one is part 2 on the function and rearrangement of immunoglobulin genes. We're looking at the specific molecular mechanisms that allow one part of the DNA to break and rejoin with another part of the DNA in order to produce this rearranged gene. So in order to do that, we're going to review the model of the structure of the antibody, the structure of the gene, and then we're going to go in and take a much closer look at the specific nucleotide sequence that allows these rearrangements to take place. And then finally, we're going to manipulate some models to give you an idea of how it works. So as a reminder, I showed you this physical model before of the kappa light chain gene. And if you will recall, here is the part for the constant region, the joining regions, and then we had a whole slew, a selection of well over 30 different variable regions, and we can pick any one of the variables and put it to any one of the joinings. So just as a reminder, what that gene will do after rearrangement is code for this part of the antibody here. The constant region will code for this part of the light chain. The variable region here in pink will code with the joining region for the variable domain. So the variable domain is coded for by the variable and joining regions, the constant domain by the constant. What we're specifically looking at here today is the part of the gene that codes for that. That is the region where the joining and variable parts of the gene get together to code for the peptide that forms one of the loops in the antibody. So let me take this down for a minute. And I'm going to just put this up here to remind you that here we've got this very, very, very long gene. Now, this gene has one and only one constant region, so it's always going to make the same instructions for the constant domain. It does, however, have a number of joining regions, each one of which is preceded by this three-part recombination signal sequence. So here's a joining region with its recombination signal sequence, and we have 5 of them, okay? We also have a huge, long series, of different variable regions, and we're going to pick one of these variable regions at random to put together with the joining region. So when we get done with this, the functional part of this gene will have one variable region here, and there will be more upstream, but we won't use them. It will be joined directly to a joining region, but remaining in between these two, and what will now be part of the coding region, will be a few of those nucleotides from one part of the signal sequence. We may have extra J's, we have the constant region here, but we do not put the constant region directly onto the joining. That's going to be a function of message processing later. So here we've got a functional light chain kappa gene, and we will see when we go into the lambda that it looks rather similar, even though we start out with a rather different product. So let's go and do one of these. Let's take a look at the recombination signal sequence. If you will recall, I do the variable gene region in pink, and here I've got the variable region that I'm going to use, and immediately downstream from this is a 7-base nucleotide sequence. It's 7 bases, so it's called a heptamer. This heptamer with the 7 bases here is a palindrome. And if you read those sequences carefully, you can see that they say the same thing whether you read them forwards or backwards. And that's palindrome, like Madam, I'm Adam, or Able I was, ere I saw Elba. In any event, it's something that reads the same forward and backwards, and that's going to be important in generating some of our loops later on. What follows after that is a 2-turn sequence, and that's simply 23 bases. What follows after this downstream is the nonomer. That's 9 bases, and it's an AT-rich nonomer, so we have lots of A's on one side, and as it turns out, lots of T's on the other. If I look at, on the other part, the joining region, I see that I have sort of the mirror image of this. Okay. I have the part that codes for the joining region here. I have next, as I go upstream, the palindromic heptamer. I have next going upstream a 1-turn sequence composed of 12 bases. And on the other side here, at the very upstream end, again, I have the AT-rich nonomer. Now, what does that mean? What are the functions? What I've got here are a series of bases on upstream of the joining region, downstream of the variable region, and the function of those bases is to bring these two DNA regions together and allow us to join them. And so we have to hold things together in the correct orientation. One thing that's very important for this is that we always join a 1-turn and a 2-turn signal sequence. And so what I've done is I brought in a ball and stick DNA model. And here I have a way of showing you what a 1-turn sequence is. So if I start out up here and I count 12 bases, because that's what you find in a 1-turn sequence, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, you can see that I've gone pretty much 1 turn around the DNA. So it takes 12 bases and you go for 1 turn. If I go another 11 more, because it's, a little over 1, I will go all the way around 2 times. And unfortunately, this DNA model has only 22 bases, but the 2-turn sequence will, you can see, be enough nucleotides so that if I were to look at the DNA that's coded for it, it would make 2 turns around the axis of the DNA molecule. Okay? 1 turn, 2 turn. When I put together 2 recombination signal sequences, or RSSs, I always put together a 1 turn with a 2 turn. In your textbook, they use a shorthand for this, and that is if it's a 2-turn sequence, here, they use a triangle to illustrate that. And the triangle has its point in this direction. Here, like this, and they use a dark blue triangle, only it's fatter than that. And for a one-turn sequence, they use kind of a light red triangle. And it goes like this. Only, of course, it's fatter than that. So when you see these triangles in your textbook, each triangle represents the sequence of nucleotides that allows these two parts of the gene to get together. So what is the exact mechanism? Well, let me go use my yarn. So here I have my yarn indicating the gene. And I'm going to pick out one of the variable regions. At random. I just sort of grabbed it. Okay. And I'm going to pick out one of my joining regions. Again, at random. Dum-de-dum. Here it is. Now, I have to line these up. And I'm going to line them up kind of like this. You can see that the recombination signal sequences overlap. And what I'll always have here to here is a 3-turn sequence of DNA. I'll have the AT-rich ones here that bring the whole thing together. And then I will have the heptamer in here that participates in actual putting these guys together. The first thing that happens here is that you make a cut in the DNA. And so happily, although it's not that easy to get apart, I've got a 2-ply piece of yarn. And so what happens first of all is that there is a cut in one side of the DNA. So I have one strand that's kind of hanging out, and the other strand is still connected. The strand that's hanging out comes around here and attacks this remaining strand and forms a hairpin loop. And so now I have a hairpin loop here, and that causes this part of the molecule there to get cut off and taken away. Okay, now maybe I can open this up a little bit, but what you should see here is that one strand of the DNA comes in, loops around this way, and loops around back into this. So if I could open this up, it would look a little bit like a lollipop, or what it would look like would be one of those things that you'd blow soap bubbles with. You'd have a sort of stick-like structure here and a loop-like structure here. Okay, the same thing happens over with the DNA of the joining region. First, something happens that allows this to open up. You cut off one strand. That strand comes around and attacks the other side, forms a hairpin loop like this here. And then that cuts this part free. Now notice, this part actually will get hooked up together. But look what I now have here. I have a part of the DNA that's being flat out removed. What I have taken out is any of the J regions that are upstream of this joining region, and any of the variable regions that are between the one I'm using and the J region. But on the other hand, and this is going to get taken away and recycled. On the other hand, if you notice, I still have an awful lot of leftover variable regions upstream. These are not removed, but they are never used, because during this process here, I will activate a promoter region in front of this leader sequence, and only this part of the gene from here on downstream will be transcribed. Now, what happens next? I have two loops here. What I'm going to do is cut those loops open again and hook them up. Now, the problem is I'm going to cut them open kind of at random, so that when I'm done, I may have a gap in here. In other words, this part of the strand, single strand, might be longer than the other complementary strand. In that case, what I'll do is I'll trim things around, and I will fill in bases in this region here so that I have a complete double-stranded DNA molecule where both strands are complementary to each other through this entire joint sequence. And so here I've got the joining region now is now contiguous with the variable region through this joint sequence. Now that's the good news. The bad news is that there's no guarantee that I'm going to have exactly 3 nucleotide combinations as I go through here. In other words, I've got codons in the variable, I have codons in the joining, and if I had groups of 3 nucleotides in the signal joint, all would be well. But there's only a 1 in 3 chance that that is going to happen. And so, at least 2/3 of the time when I do this, I make what is called an unproductive rearrangement. That unproductive rearrangement means that I've screwed up, and the cell will then try the other kappa gene, and if it does screws up again, it'll try the other lambda gene, and then the final lambda gene, and if nothing works, it will undergo apoptosis. So a lot of times we have situations where this rearrangement screws up, and it results in the death of the cell. Okay, why waste this? Why not arrange this so that all the time it comes out right? Because we can certainly do that with splicing of messages. Well, it turns out that this region codes, if you'll recall, for that loop in the variable chain, and I add randomness to my antibody by fooling around here at this joint. That is, there is more than one way to cut and rejoin the various nucleotide sequences in here. And so what I've done is I've done a model that basically shows how this can go in 3 different ways. All right. In other words, I might cut this one here and this one here, and I would have a huge long piece on the bottom, like this, and I would have to do a whole lot of P nucleotide filling in here to make them come out even. I could do something in the course of this that got rid of most of this. Here and here. And I could trim a few more, and I could do very little P nucleotide addition, or maybe none. And there. And you can see I could have other ways to do it here. Okay, I could have something that basically came out even, like this, because I did it long on one strand, short on the other. Long on one strand, short on the other. And so there might be much less nucleotide addition. But the point is that as I cut this thing open, ligate it, and fill it in, I am basically tossing the dice at this junction. And when I toss the dice, I may wind up with a dead cell, but on the other hand, I may wind up with a brand new combination of amino acids in that loop, and that will give me a unique opportunity to identify and get rid of a pathogenic epitope. Okay, so this is the process for the light chain. When we look at the one for the heavy chain, it will be very similar, except that we will also do some additional nucleotide addition in here called N-nucleotide addition. But before we do that, we're going to, in part 3, take a closer look at the lambda gene. So this is it for recombination in both light chain genes, and we'll look at a comparison of this and the lambda gene in the next clip.